Hello everyone, my name is Ahmad Alftieh and today I will be presenting our research findings for a study titled Enlarged Pituitary Gland Volume, a possible state rather than trait marker of psychotic disorders. Psychotic disorders are among the leading contributors of negative life outcomes and trajectories. Enlarged pituitary gland is observed in the development of psychosis and could be a useful biomarker. However, studies investigating pituitary gland volume in psychotic disorders report mixed findings. Therefore, we sought to determine whether people with psychotic disorders have larger pituitary gland volumes compared to healthy controls and to investigate the clinical associations of pituitary volumes. We hypothesized that individuals on the psychosis spectrum would have larger pituitary glands than healthy controls, and that enlarged pituitary glands will be associated with greater severity of clinical symptoms, shorter duration of illness, higher level of cognitive impairments, and higher dosage of antipsychotic treatments. A total of 751 participants were included in this study. 174 individuals had schizophrenia, 114 individuals had schizoaffective disorder, 167 individuals had bipolar disorder, and 296 individuals were healthy controls. These participants were recruited from the Bipolar and Schizophrenia Network for Intermediate Phenotypes, which is a network of investigator-driven laboratories focused on developing phenotypes, genotypes, and biomarkers for psychosis. Participants engaged in full clinical characterization, which included brain imaging assessments and electrophysiological assessments. Participant demographics such as sex, age, race, duration of illness, and antipsychotic dosage were recorded. In order to measure the volume of the pituitary gland, we employed a label fusion technique called MAG-ET brain algorithm to automatically segment the pituitary gland. This method has been previously validated to segment the pituitary gland, showing high spatial overlap between manual segmentation of the pituitary gland volume and their respective automatically generated labels. This figure displayed is an example of the pituitary gland segmentation conducted on one of the study participants. Moving on to our results, Figure 1 shows between group comparisons on pituitary gland volume. Figure 1a shows all the patient groups compared to controls. Although the patient groups had a larger pituitary gland than controls, the differences were not significant. Figure 1b shows comparisons between each of the patient groups. We found that individuals with schizophrenia seem to have a larger pituitary gland volume, however, the differences were also not significant. Furthermore, post-hoc pairwise comparisons showed no significant differences in pituitary volume between the patient groups compared to controls or between any of the patient groups themselves. Lastly, we looked at the associations between factors related to psychosis and pituitary gland volumes in individuals with psychotic disorders. We looked at cognitive impairments as indicated by the brief assessment of cognition in schizophrenia. We looked at symptom severity as indicated by the positive and negative syndrome scale. We also looked at antipsychotic medication intake as indicated by CPZ equivalence. And lastly, we also looked at the duration of illness. Our findings showed that only symptom severity seemed to have a significant association with pituitary volume, as shown in Figure 3b. So in conclusion, we found that individuals with psychotic disorders do not show significant differences in pituitary volume compared to healthy controls, contrary to our hypothesis. We also found that symptom severity is the only variable that has a significant impact on pituitary volume among individuals living with psychotic disorders. It is also noteworthy to mention that this study is the first to investigate pituitary volume in a large sample of individuals with transdiagnostic psychotic disorders. So what does all of that mean? Our results suggest that a larger pituitary gland volume is not a trait biomarker for psychosis, but may serve as a state-related biomarker linked with severe symptoms. This helps clarify previous inconsistent reports and supports further investigation of stress vulnerability biomarkers in psychosis. Lastly, future longitudinal studies should seek to determine if increased pituitary volume correlates with increased symptom severity over time in individuals with psychosis. Thank you for listening to my presentation. I'd like to acknowledge that this research is supported by FRQS and ERM-H awards, which has been granted to my supervisor, Dr. Cynthia Guimon.